Hi everyone, I hope you all are doing well. I am Sanjay Lad. Welcome to one more video of Special BIO-TONIC. In today's session, we'll talk about molecular basis of inheritance, that is structure of DNA. Students, I request you all to like my channel, share it and subscribe it, and don't forget to press bell icon. So let us start with structure of DNA. Students, nucleic acids like the DNA and RNA comes under nucleic acid. So nucleic acids are of two types. One is RNA and second is DNA. Now these nucleic acids are made up of nucleotides. Now we call the DNA and RNA as polymer of nucleotides. Now these nucleotides are made up of these three basic components. One is nitrogen base, second is sugar, and third is phosphate. Students, nitrogen bases are of two types. One is purines, second is pyrimidines. The sugar are of two types, ribose sugar and deoxyribose sugar, whereas phosphate is phosphoric acid. Now, purines are of two types, one is adenine and second is guanine, whereas pyrimidines are of three types, cytosine, thymine and uracil. Now, ribose and deoxyribose sugar are pentose sugar. So pentose means the sugar containing 5 carbons. So molecular formula of ribose sugar is C5H10O4, whereas deoxyribose sugar is C5H10O4. So in deoxyribose sugar there is one oxygen less as compared to ribose sugar, whereas phosphoric acid is written as H3PO4. Now here A, G, C, T, U, these are the symbols of these nitrogen bases. You must be aware of these symbols. Now ribose sugar is found in RNA, whereas deoxyribose sugar is found in DNA, and phosphoric acid is written with P with circle. Now students, we know that adenine pairs with thymine in case of DNA, whereas in case of RNA adenine pairs with uracil, whereas guanine pairs with cytosine. So I hope you are understanding the basic biochemistry of nucleic acids or nucleotides. Now when we talk about DNA, DNA is double stranded, whereas RNA is mostly single stranded, but part of it can become double stranded. Now with this information, what I want to say is that due to complementary base pairing, if you know one strand of DNA, you can write the other strand of the DNA because of complementary base pairing. Now what is the meaning of complementary base pairing? That we know that A always pairs with T, C always pairs with G. So if you know one strand of DNA, and you can write the second strand easily by like say A pairs with T, T pairs with A, like this. Same is the case with RNA. If you know one strand of RNA, you can write other strand of RNA easily because of complementary base pairing. Only difference is that in case of RNA, thymine is replaced by uracil. Please remember this. Now here, these are the nitrogen bases present in DNA, whereas these are nitrogen bases present in RNA. I'll tell you a trick to remember nitrogen bases of DNA and RNA. Suppose, say, wife of police wala want to hand over, say, vessel, that is shitty we call, to her husband. So what she will say, AG city leke jana. What she will say, AG city leke jana. And while going, she will say AG CU. So remember, AG CT is what? Nitrogen bases in DNA. AgCu is nitrogen bases in RNA. So these are the tricks or mnemonics you can remember while answering regular board exam or NEET examinations. Now these nucleotides are made up of two types depending on the sugar present. Ribonucleotides contain ribose sugar, whereas deoxyribonucleotides contain deoxyribose sugar. Nucleotide we have seen in the previous slide that they are made up of nitrogen bases, sugar and phosphate. Same is the case with this, nitrogen bases, sugar and phosphate. So I'll tell you one more mnemonic to remember these three elements. BSP, that is border security police, border security police, base, sugar and phosphate. So what are the elements in nucleotide? Base, sugar and phosphate. Okay, now nitrogen bases in the RNA are, we just now told, AGCU. Sugar is ribosugar. Phosphoric acid is common in both DNA and RNA. Nitrogen bases in DNA are AGCT. Sugar is deoxyribose, whereas phosphate is same. Now, what is the structure of a nucleotide? Nucleotide is made up of this central element called as, say, sugar, which is ribose here. Now, phosphate is attached to sugar at 5 carbon, whereas base is attached to sugar at first carbon. Now, few more things we want to learn here, that this ribose sugar is a pentose sugar. It contains 5 carbon. Now, out of 5 carbon, say first, second, third and fourth, these 4 carbons are present in the ring, whereas fifth carbon is away from ring. So please remember, first 4 carbons are there in the ring, whereas the last one is away from ring. Now base is attached to sugar at first carbon, whereas phosphate is attached to sugar at fifth carbon. So a nucleotide is made up of base, sugar and phosphate. Border security police, remember this mnemonic. Now one more thing I want to tell you, that the numbering is said as 1 prime. This dash indicate prime. So this carbon is called as 1 prime, this carbon is called as 2 prime, 3 prime, 4 prime and 5 prime. So remember this notation also. So this is a nucleotide which contain ribose sugar, therefore this is ribonucleotide. Now the second is deoxyribonucleotide. Nucleotide is made up of base, sugar and phosphate. Now base is attached to sugar at this 1 prime carbon, whereas phosphate is attached to sugar at 5 prime carbon. Please remember this. So I'll repeat again. Ribose sugar is present, therefore this nucleotide is called as ribonucleotide. This is having deoxyribose sugar, therefore this is called as deoxyribonucleotide. Now RNA is a polymer of ribonucleotides. RNA is a polymer of ribonucleotides, whereas DNA is made up of deoxyribonucleotide. Now there's one more term here, that is nucleoside. So base plus sugar is called as nucleoside. I'll repeat again, base plus sugar is called as nucleoside. There is a word ending with SIDE, side, nucleoside. So base plus sugar is nucleoside, whereas base plus sugar plus phosphate is called as nucleotide. I hope you are understanding difference between ribonucleotides and deoxyribonucleotides. Now when we talk about the structure of DNA, now this is the structure of DNA. Now for the sake of understanding, I have written these 4 deoxyribonucleotides one above another. Now here the first base taken is adenine, cytosine, thymine and guanine. Now here adenine is attached to this deoxyribose sugar at first carbon, whereas the phosphate is attached to sugar at fifth carbon. Same is the case with all nucleotides. Now as I said just now, DNA is a polymer of deoxyribonucleotide, and these deoxyribonucleotides are joined by phosphodiester bond. Now this phosphodiester bond is formed between 5 prime carbon of second nucleotide, which is attached to 3 prime hydroxyl group of previous one. I'll repeat again. The 5 prime phosphate of second nucleotide is attached to 3 prime hydroxyl group of the first nucleotide by this phosphodiester bond. So this white bond which you see is a phosphodiester bond. Now second is, this is a phosphodiester bond between these two nucleotides. Then this is a phosphodiester bond between these two nucleotides. So DNA is a polymer of deoxyribonucleotides which are joined together by phosphodiester bonds. Now the other strand of the DNA is anti-parallel. They are upside down. Now here, as we know that adenine pairs with thymine, so that's why thymine is taken here. Now the structure remains same, barring that only thing is the other strand of the DNA is anti-parallel, that is upside down. Now in second case, this is a phosphodiester bond formed between these two nucleotides. Then this is a second phosphodiester bond, and this is third phosphodiester bond. Now in DNA we know that adenine pairs with thymine with two hydrogen bonds. Remember, this is hydrogen bond. Phosphodiester bonds are covalent bonds which are difficult to break. Hydrogen bonds are easy to break. Now cytosine pairs with guanine with three hydrogen bonds. So AT pairs with thymine with double bond hydrogen bonds, and C pairs with G with three hydrogen bonds. So I hope you are understanding molecular structure of DNA. Now if you know the sequence of this strand, the head of this nucleotide is 5 prime, whereas the tail of this is 3 prime. So the total direction of this left DNA strand is 5-3-. It is written as 5-3- because its head is ending with 5 prime and its tail is ending with 3 prime. Its tail is ending with 3 prime. So therefore one strand is 5-3-, whereas in case of second, the tail is 3 prime whereas head is 5 prime here. So it is 3-5-. So here the DNA is made up of two strands, and these two strands are anti-parallel. They are opposite directions. Please remember this. Then it is made up of nucleotides, precisely deoxyribonucleotides, joined together by phosphodiester bond, and nitrogen bases are joined together by hydrogen bonds. Please remember this. Now with this information, we'll see the structure of DNA proposed by James Watson and Francis Crick. Students, structure of DNA was proposed by James Watson, Francis Crick, and Maurice Wilkins. There was one more lady, Rosalind Franklin. So her name is not written here. Now what they said is that DNA is made up of two strands. So DNA is double stranded and the two strands are opposite of each other. Therefore it is written as 5-3- and second is 3-5-. So one strand is going from 5 end to 3 end, that is 5-3-, whereas other is going from 3- to 5-. So DNA is double stranded and they are anti-parallel. Now the backbone of the DNA is made up of sugar and phosphate. This S stands for sugar whereas P stands for phosphate, whereas the steps of the ladder are made up of nitrogen bases. Now nitrogen bases are of two types, purines and pyrimidines, and just now we said like purines are of two types, adenine, guanine, whereas pyrimidines are cytosine and thymine, that we know. Now A pairs with T with two hydrogen bonds, whereas C pairs with G with three hydrogen bonds. Now one turn of the DNA has 10 base pair with the length of 34 angstrom. Therefore the distance between successive nucleotide is 3.4 angstrom. So here one turn contains 10 base pair with 34 angstrom. Therefore length of one base pair is 3.4 angstrom. This, if you convert angstrom into nanometer, this becomes 34 nanometer, whereas this becomes 0.34 nanometer. So just remember, angstrom is smaller as compared to nanometer. Now because of twisting, the DNA shows the large grooves called as major grooves. Major means large, and small grooves are called as minor grooves. So I hope you are understanding the structure of DNA. Now the diameter of the DNA is 20 angstrom. So I'll repeat again all the points. The DNA is double stranded and anti-parallel. Why it is called anti-parallel? Because one strand is 5 to 3, second is 3 to 5. Then the backbone of the DNA is made up of sugar and phosphate, whereas the steps of the ladder are made up of nitrogen bases. Nitrogen bases are of two types, purines and pyrimidines. In purines are adenine, guanine, whereas pyrimidines are cytosine and thymine. Then A pairs with T with two hydrogen bonds, whereas C pairs with G with three hydrogen bonds. Remember, these are hydrogen bonds. Now one turn of the DNA contains 10 base pair with a length of 34 angstrom. Therefore, the length of one base pair is 3.4 angstrom. Due to twisting, DNA shows major grooves and minor grooves. Large grooves are called as major grooves, whereas small grooves are called as minor grooves. And the diameter of the DNA is 20 angstrom. So this is a structure proposed by Watson, Crick and Morris Wilkins, for which they got Nobel Prize. Students, I hope you are understanding structure of DNA. I highly recommend you to draw these diagrams and make a separate drawing book in which you can draw these diagrams. And at the end, I request you all to like my channel, share it and subscribe it. And subscription is totally free. So don't forget to press bell icon. And thank you for watching this video.